Hello students, in this class we are going to discuss about the prokaryotes. Basing upon the organization of DNA, the cells are of two types, prokaryotic cell and eukaryotic cell. The organisms which are made up of prokaryotic cells are called as prokaryotes, and the organisms which are made up of eukaryotic cells are called as eukaryotes. So prokaryotes are usually single-celled organisms. They have no definite nucleus. There is no organized DNA and there is lack of cell organelles. Membrane bound cell organelles like mitochondria, endoplasmic reticulum, Golgi body, etc. But in eukaryotic cells there is a definite nucleus. It is having a boundary which is called as nuclear envelope. There is organized DNA inside that nucleus and also membrane bound organelles like mitochondria, endoplasmic reticulum, etc. are there inside a eukaryotic cell. In this class we will only discuss about the prokaryotic cell, which are usually single celled organisms. The term prokaryote has come from two words. Pro means primitive and karyon means nucleus. So these are having a primitive type of nucleus. Actually there is no definite nucleus in the cytoplasm. Only a nucleus like region in the cytoplasm where there is naked DNA or chromosome is found naked, and there is no membrane bound organelles in a prokaryotic cell. So first we will list out the components which make a prokaryotic cell. Later we will discuss one by one. So what are the components which make a prokaryotic cell? Number one, it is nucleus and nuclear material. Then the plasma membrane, cell wall, then glycocalyx or capsule, or it is called as slime layer, then the appendages. Appendages are the protrusions from the cell surface. Next to appendage there are mesosomes, ribosomes, then photosynthetic thylakoids, then gas vacuoles and respiratory enzymes. So these components make a prokaryotic cell, but not always it is necessary that all components should be present in a definite prokaryotic cell. So let us draw a diagram. This is the typical structure of a rod-shaped bacterium. We are not discussing here the exact structure of bacteria here. The bacteria are coming under the prokaryotes. So I have taken here one bacterium, one rod-shaped bacterium. Later in next class we will study detail about the structure of bacteria. So this is the plasma membrane which protects the cytoplasm and intracytoplasmic substances. In most of the bacteria, the plasma membrane is made up of phospholipid bilayer as that of the eukaryotic cells. But in archaea, which is another domain of prokaryote or other group of organisms which are coming under the prokaryotes, they contain phospholipid monolayered plasma membrane. So this is plasma membrane which protects the intracytoplasmic substances. Next to plasma membrane there is the cell wall. This is the cell wall. Outer to plasma membrane the cell wall, which is not necessarily present in all prokaryotic cells. It is made up of peptidoglycan. Mycoplasma do not possess cell wall, but all the bacteria, they possess cell wall. So what is the function of this additional cell wall? This provides definite shape to a prokaryotic cell, and also the cell wall prevents the cell from bursting out. When the prokaryotic cell is treated in a hypotonic solution, then it takes up lots of water. So there is possibility for bursting of the prokaryotic cell. But due to the presence of this cell wall, the prokaryotic cell does not burst. So it prevents bursting of cell. It protects. So next to cell wall, there is one more additional layer outer to the cell wall, the gelatinous layer, which is made up of mainly glycoprotein. The gelatinous layer which is made up of the glycoprotein. This is the glycoprotein layer, which is called as capsule or slime layer or glycocalyx. It prevents the cell from drying out or desiccation, and also it helps to cling to other prokaryotic cell and helps it in attachment with the other prokaryotic cell. Suppose here another cell, bacterial cell, and the outer cell wall here and the gelatinous layer here, the capsule. With the help of this gelatinous material, the prokaryotic cells are attached with each other, or they cling to each other. Next to it, one more structure. In the cytoplasm, there is naked DNA or naked chromosome. That region of the cytoplasm which contains that naked DNA is called as nucleoid or genophore, and this is the chromosomal DNA. Sometimes in some bacterial cell, in addition to this chromosomal DNA, there is more number of extra chromosomal double-stranded DNA, which are called as plasmids. So next we'll draw another structure, the ribosomes, which are freely floating in the cytoplasm. These granules are called as ribosomes, which are of 70S type. S stands for Spethborg unit. So these are smaller ribosomes. Next, there are the infoldings of the plasma membrane. Which are called as mesosomes. These are called as mesosomes. Mesosomes usually contain respiratory enzymes. So they help in the process of respiration, and it is the function of the mesosomes is similar to that of the mitochondria in eukaryotes. These are similar to mitochondria. It also helps during the process of conjugation, the mesosomes. Next, there are appendages. Mainly there are three types of appendages. Appendages are the protrusions coming out of the cell surface, which usually helps the cell in locomotion. Or that is the movement of the cell from one place to another, for attachment with the surface or substratum or with other cell, and reproduction in the exchange of genetic material. So there are three kinds of appendages seen. Number one, the flagella. Singular is flagellum. These are very few in number, the flagella, and they are long. The microtubules inside the flagella, they are arranged in 9+2 form. The continuous thrust or lashing movement of flagella, like sailing a boat, the flagella helps the prokaryotic cell to locomote or to move from one place to another. So these are few in number. So there is one more type of appendage, the pili, or also called as sex pili. Singular is pilus, Pilus. These help in the exchange of genetic material during the process of conjugation. These are made up of tubules. Actually, these are tubular structure. These are tubular and broad structure. Tubular and broad. And usually 1 to 4 pili are present in a prokaryotic cell. They help in exchange of the genetic material between the prokaryotic cell during the process of conjugation. One more type of appendage, which are numerous, present throughout the surface of the prokaryotic cell. They are called as the fimbriae. These are the fimbriae. They are shorter than flagellum. These are fimbriae. (300 to 400 fimbriae are present in one prokaryotic cell. They help in the attachment of the prokaryotic cell with the substratum. With the help of this fimbriae, the prokaryotic cell get attached with the substratum. That means it may be the rock or any surface, any sediments under the sea. With the help of this fimbriae, it gets attached. Next, we'll move to the thylakoids. In some photoautotrophic prokaryotes, for example, blue-green algae, and some photoautotrophic bacteria, thylakoids are present scattered in the cytoplasm. In eukaryotic plant cells, the thylakoids are arranged organized inside the chloroplast. But here in photoautotrophic prokaryotic cells, the thylakoids are scattered in the cytoplasm. These are thylakoids. Which help in the photosynthesis. Next, gas vacuoles are present. Gas vacuoles. These are the gas vacuoles. Next, respiratory enzymes. Respiratory enzymes can be found on the surface of the plasma membrane, and also they are found inside the mesosomes. These are the respiratory enzymes. These are the respiratory enzymes. The translation and transcription process all occurs inside the cytoplasm of the cell. So this is all about the brief structure of a prokaryotic cell.